Well, I posted a lot of articles that have talked about new therapies that are targeted at repairing myelin. But if we start taking one of those someday, how will we know if it's working? Well, hello, and welcome to another episode of You and Me and Multiple Sclerosis. My name is Pam. I've been living with multiple sclerosis for over 40 years. And even though I'm not able to be in my usual spot today, I'm continuing to be out there looking at interesting developments in the world of MS so that I can share them with you. And I found a couple of interesting articles that I want to look at today. But in reading those articles, I have realized that having a new therapy is not enough. You need the diagnostic tools that allow you to tell whether or not that therapy is doing any good. And it's certainly not having to wait years to find out if it's doing any good. Let's take a look at these two articles, and you can see the direction things are heading. The first article is in Multiple Sclerosis News Today, and it was published on February 7th of 2024, so it's not quite as new as some of the articles we look at, but it's quite relevant. I think it's worth looking at. It's called New MRI Technique Allows for More Precise Imaging of Myelin Sheath. Approach overcomes key barriers to directly mapping myelin in MS patients. Scientists have developed a technique to more precisely map the myelin sheath, the fatty covering around nerve fibers that is damaged in multiple sclerosis, in people undergoing an imaging scan. While further work is needed, its use produced promising initial results, the scientists wrote, noting that this approach, once optimized and embraced, could allow for a more accurate MS diagnosis and better monitoring of myelin damage and repair in MS patients. So as you can see here, the two key things they're looking for are diagnosing MS in the first place and also better monitoring of the myelin damage, but also, I would hope, any repair strategies that are employed. They say that the method was described in the study Myelin Bilayer Mapping in the Human Brain in Vivo, published in Magnetic Resonance in Medicine. Current MRI technology cannot directly image the myelin sheath in MS patients. MS is caused by inflammation in the brain that damages the myelin sheath, leading to problems with nerve signaling and ultimately giving rise to disease symptoms. To monitor myelin damage in patients, researchers and clinicians for decades have relied on magnetic resonance imaging, or MRI. MRI basically works by using powerful magnets to generate a signal from hydrogen atoms. Specifically, an MRI detects signals from hydrogen atoms in water, which is 2 parts hydrogen and 1 part oxygen. Because a traditional MRI detects water, it cannot directly measure myelin, made up of fats and proteins. Instead, it indirectly estimates myelin amounts by measuring water between the layers of myelin. These fats and proteins also contain plenty of hydrogen atoms, so it's theoretically possible to directly image myelin itself during an MRI. But this technically hasn't been achievable, largely because signals from hydrogen and myelin are much briefer than signals from water, and a conventional MRI scanner is unable to take measurements quickly enough to detect them. Put simply, the hydrogen atoms in myelin tissue move less freely than those in myelin water. That means they generate much briefer signals, which disappear again after a few microseconds. And bearing in mind that a microsecond is a millionth of a second, that's a very short time indeed. Marcus Wieger, PhD, co-author of the study at ETH Zurich in Switzerland, said in a university news story. The researcher's technique involves a specialized MRI scanner that specifically analyzes a patient's head and contains especially powerful magnets, allowing faster detection of signals. The greater the change in magnetic field strength generated by the three scanner coils, the faster information about the position of hydrogen atoms can be recorded, said Emily Bodsvik, a researcher at ETH Zurich and the study's first author. Further refinement needed, but accuracy seen in proof of concept tests. The new system is not without limitations. In particular, scans take about an hour and a half, which the researchers said is at the upper limit of what cooperative volunteers can be expected to manage. Well, I don't know about that. The last MRI I had was 3 hours, and I had to cooperate for that. So I think an hour and a half sounds like a picnic by comparison. Anyway, still, they showed proof of concept experiments that their system could accurately image the myelin sheath in healthy adults with a resolution of less than 2 millimeters. Researchers now plan to further refine the technology and test it on MS patients, and they ultimately hope to work with industry partners to bring the technology into clinical use. Myelin bilayer mapping has the potential to meet the myelin monitoring needs of clinical and research communities, be it for applications in basic research, diagnosis, disease monitoring, or drug development, the scientists wrote. We've shown that our process works, Wexler said. Now it's up to industry partners to implement it and bring it to market. Well, I see that development is interesting. I'll keep an eye out to see if any industry partners have stepped up to the plate. I haven't heard about that yet, but I think that the interest for us would be that with all this development of myelin repairing drugs that is in the pipeline, the best way to know whether the drugs are working is if you have some good diagnostic tools. And this might very well be a good diagnostic tool to see whether your myelin is actually being repaired, or at least if the damage has been halted. The next article I saw is much more recent. It was published on August 12th of 2025. It also involves a very familiar diagnostic tool being applied to the concept of being able to actually see what's going on with our myelin. The title of the article is New PET Tracer Imaging Tech Could Help Track Nerve Fiber Loss in MS. Study. It may provide means to select candidates for treatment and monitoring. That would be really neat. A new positron emission tomography or PET tracer may help determine the extent of nerve fiber loss in multiple sclerosis lesions, something that can't be identified with conventional MRI imaging, according to a new study. So interesting. It's like these two are going head to head now. Let's see which one actually gets developed. Researchers think the new imaging tech could help track the efficacy of experimental therapies that aim to promote myelin repair. Precisely. That's what I got excited about when I saw that. The study, first evaluation in multiple sclerosis using PET tracer, 18F 3F4AP, demonstrates heterogeneous binding across lesions, was published in the European Journal of Nuclear Medicine and Molecular Imaging. PET tracer highlights axons that have lost myelin. In MS, inflammation in the brain causes damage to the myelin sheath, which is a fatty covering that wraps around axons or nerve fibers and helps nerves to send electrical signals. The loss of myelin leads to axonal dysfunction and damage, disrupting normal nerve signaling and ultimately driving MS symptoms. MRI scans are a standard type of imaging technology used to track disease activity in MS. On MRI scans, areas where myelin has been damaged and lost are visible as spots called lesions. A drawback of MRI scans, however, is that they cannot differentiate lesions with intact axons versus lesions where axons have been lost. MRI images the body's structures by detecting signals from water molecules. PET scans are an imaging technology that generally use tracers, which are basically molecular dyes that stick to specific biological structures and cause them to light up on the scans. Studies in animal models have indicated that a novel tracer called 18F3F4AP can highlight axons that are intact but have lost myelin. The tracer specifically binds to certain potassium channels that are normally hidden under intact myelin but exposed on nerve fibers when myelin is lost. It is developed by radioactively labeling the active ingredient ampera or delfambardine, a therapy approved to improve walking ability in people with MS. They say that results in humans are largely consistent with those in preclinical animal studies. An ongoing phase 1 study, and there's the number, is assessing whether the tracer can be used to detect changes in myelin. The study is a joint effort between Quantum Biopharma and Massachusetts General Hospital. Quantum hopes the tracer will be able to show in a follow-up trial that its drug Lucid MS, which we just talked about last week, can prevent demyelination and promote myelin repair. The ongoing collaborative study with MGH seeks to further evaluate the imaging agent and its potential to demonstrate the effectiveness of drugs such as Lucid 21302 or Lucid MS that can protect the myelin sheath in MS. Andrei Trzynski, MD, PhD, Vice President of Scientific and Clinical Affairs at Quantum, said in a company press release. The researchers shared results from 3 people with MS and 3 people without the disease who were enrolled in a phase 1 study. Their goal was to determine if the tracer could distinguish between different levels of myelin and axonal loss. In this study, we first evaluated the robustness of 18F3F4AP, boy, they need a nicer name for that, as a brain imaging agent in humans, including people with MS, the scientists wrote. Participants underwent standard MRI scans as well as PET imaging with the novel tracer. Tracer uptake in the brain was generally higher in MS patients, which was expected given that the tracer is thought to highlight myelin loss. The tracer also showed good consistency from person to person, the researchers said. The relatively low variability across subjects suggests this tracer meets the requirements to become a robust biomarker, the researchers wrote, noting that the results in humans were largely consistent with prior results in preclinical animal studies. Among the MS patients, some lesions showed higher than normal levels of the tracer on PET scans, but other lesions had lower than normal tracer binding, and still others had normal tracer binding. I wonder what that all means. Well, good, it looks like they're going to tell us. The researchers proposed that lesions with high signals likely represent areas with myelin loss, but where axons are largely intact, whereas low signals indicate axons that have been lost. Normal signals, meanwhile, may indicate relatively little myelin loss or some myelin repair. Perhaps the most remarkable finding of our study, which, if confirmed, could change the way we understand and monitor the heterogeneity of lesions in MS, was the different binding of the tracer across MS lesions, the researchers wrote. The scientists emphasized that further studies are needed to validate the results and confirm if their interpretation is correct. If it's true that this tracer can distinguish lesions with and without axon damage, then it may provide a means to select candidates for remyelinating treatments and monitoring for the effect of those treatments, the researchers wrote. Quantum is now working to launch a phase 2 trial of the therapy in people with MS. The company expects to file for clearance of the trial later this year. The published study shows that the PET tracer is highly promising as a biomarker to detect and monitor lesions in people with MS, Brzezinski said. I think there's a couple of things that I learned, at least, from the articles today. Number one, I got a better explanation of how MRI works and how PET scans work. And they're not exactly the same, obviously. They've got some big differences in how they can diagnose what's going on or monitor what's going on. And both of them could be very useful. They could even be complementary. But either one of them, I think, would be a really good thing to use if you're trying to figure out whether your drug that's supposed to repair myelin loss, or at least prevent more of it from happening, whether that's actually working in real time. Otherwise, you have to rely on the subjective report from your patient of whether or not their symptoms are better or worse, which doesn't really tell you much because there are so many confounding factors with MS, you just can't know. So these two potentially very promising technologies for diagnosis and monitoring, I think, are very useful for us as we consider all of these myelin repair DMTs that are coming down the pike, and we get interested in those and we get looking forward to them. But if there's no tool to diagnose how they're working and to monitor their effectiveness, it's really not very useful. So these two articles then are helping us to see how diagnostics and therapeutics have to work together to get us the best results. Well, that's all I have for you today. I hope you found this information useful and interesting. But as always, your number one job is to take really good care of yourself, as I am attempting to do myself. Every day I'm getting a little better, so one of these days you may see me back in my old spot. But in the meantime, keep on taking good care of yourself, and I will see you again in my next video.